Now let's take a look at problem 15.24. So 15.24. Now this is an exciting, extra spicy problem. And we are, so this is I think a kind of difficult problem to explain. So if you look at the lecture slides from lecture 16, so there are 4 or 5 slides there which walk you through this problem carefully, but I'll try and do my best and go through it with a little YouTube walkthrough for you guys. Again, so here's a table of what that we'll need to understand the problem. So what happened here? Okay, here we are. Okay, so presented here are hypothetical findings from studies of heterokaryons formed from 7 human xeroderma pigmentosum cell strains. So 7 different patients. Scientists isolated, say, fibroblast cells from these patients and performed, made some cell cultures from them and are performing some studies of these cells. And so in the table, these data are measurements of the occurrence or non-occurrence of unscheduled DNA synthesis. So unscheduled DNA synthesis, UDS. So in the fused heterokaryon, okay, so they're going to take these cells and they're going to make heterokaryons from those. So we started to talk about this on Thursday. Now that was October 28th. So fibroblast cells sometimes, in culture have these pointy edges, and they're often diagrammed like this, so I will diagram them. And so let's say we have a nucleus here, and I will just I'll name the nucleus XP1. Okay. Because that is a cell from the XP1 patient, and we're calling that cell line XP1. Okay, and I'm going to call this one XP2. So this is a fibroblast cell from xeroderma pigmentosum patient, and we're calling this patient 2, and we'll call this cell line XP2 cell line, and I've labeled that nucleus XP2. So there is a technique which is really interesting. So it is called, okay, these are somatic cells, right? They're not germline cells. So somatic cell, that's easy. And if you want to fuse them together, another word for that is hybridize. So somatic cell hybridization. So somatic cell fusion. And we're going to call it somatic cell hybridization on an exam or something like that. So just know it's just fusing 2 cells. And we've got our fused fibroblast cell here with 2 different nuclei. One XP1 and one XP2. Okay, so this thing right here is called a hetero carrion. So a heterokaryon is just a cell with 2 different nuclei. At least 2 different nuclei. So 2 different nuclei. Okay, so this is pretty neat. And so one of the things we should point out is, okay, we have a set of chromosomes in here, right, from the XP1 patient. We have a set of chromosomes in here from the XP2 patient. So these can make messenger RNAs from all of the genes in there, and these can make messenger RNAs and from there, and then the proteins from genes in both nuclei are going to be made in the cytoplasm, and they're going to be shared by that whole cell. Okay, so you got 2 nuclei working together to make the proteins for the cell. So that's pretty neat, right? And so it turns out you can learn a lot of things about genes with these heterokaryons. And so what have we already learned? What have we already learned about xeroderma pigmentosum? So we have already learned that this genetic disorder is caused by a defect in nucleotide excision repair. Nucleotide excision repair, or NER, a DNA repair pathway. So, and we've also learned that nucleotide excision repair requires many different genes. So many different genes to function correctly. So, and in all of the examples I've seen, mutations that disrupt this pathway are recessive mutations. So if I am an individual, let's say I am patient XP1. Okay, so I have a gene A. There's a gene A. Gene A is required for nucleotide excision repair. So let's say the dominant one, big A here, is required to make a functional protein, right? Let's say a mutant version, little a here, is makes a non-functional protein. So an patient XP1, which might be me, okay, let's say I don't have this condition, but let's say I did, and for gene A I had 2 mutant versions. Okay, well then I would have the genetic disorder. So for some reason, there's a gene A. It's required for nucleotide excision repair. I have 2 mutant alleles for this gene, and as a result, nucleotide excision repair pathway doesn't work in me, and I have xeroderma pigmentosum. So now that's just one gene. Gene A is just one gene required for nucleotide excision repair. Okay, there could be another one. Gene B, right? Okay, let's go. Okay, so there's another patient here, XP6, say. So XP6 is a patient. We get some cells from there, and okay. So it turns out XP6 is mutated in a different gene required for nucleotide excision repair. So this patient is mutated in gene B. So instead of having either 2 wild-type copies that function normally, or 1 wild-type copy and 1 mutant copy, this patient has 2 recessive copies of gene B. So, and this patient, as a result, can also not perform nucleotide excision repair. Let's say gene A is involved with recognizing damage. So it recognizes the damage. And gene B is involved with, say, recruiting repair proteins. So 2 different functions. Both of these are required for nucleotide excision repair. If you mutate either of these genes, you lose the function of either one, completely lose the function of either one, because you have 2 mutant alleles. The process isn't going to work. So both of these individuals, even though they have mutations in different genes, cannot perform nucleotide excision repair. So in all likelihood, patient XP1, which I said was me, right? So I would have a normal for B. I should be normal for B, because xeroderma pigmentosum is kind of a rare disorder. So, you know, if you have it, you're just really unlucky, right? So your other genes are most likely going to be the wild type version. So for gene B, I'm probably normal. And XP6, well, probably normal for gene A. So what would happen if we made a heterokaryon of XP1 and XP6? So here's our heterokaryon here. Fibroblast cells, so we have the pointy edges. XP1 took my cell here. I have my XP1 nucleus fused with nucleus from XP6 individual through the somatic cell hybridization of our cells. So 2 nuclei, and so my XP1 here is making those little a messenger RNAs, which are useless. They're mutant. They don't make the functional protein. And this XP6 over here is making the little b messenger RNAs, right, from the B alleles, and XP6 is mutated in gene B. So these messenger RNAs are useless. If they're making them at all, they're useless. They're making proteins which can't participate in nucleotide excision repair. Now, however, I have normal Bs, right? So I'm making messenger RNAs for my B alleles, and okay, those are functioning great. Okay, so those are making the protein, those are being translated to a functional B protein. So it doesn't matter the XP6 isn't doing his job here, or her job, because I'm making B. And the same thing, XP6, well, this nucleus has functional A alleles. So it's making the messenger RNA from those functional A alleles, and okay, it's making the big A, those are being translated to the big A proteins, and they function. And so these big A proteins can go into the XP1 nucleus, they can go into the XP6 nucleus, it doesn't matter. We're getting lots of big A proteins that are functional from the XP6 nucleus. The same thing for B. XP1 here is making lots of functional B protein, and they can go into there, they can go into here, it doesn't matter. The whole cell is sharing the functional A's from XP6 and the functional B's from XP1. So what is the result of this? Well, it turns out if you have xeroderma pigmentosum and you take some cells from, well, you donate some of your cells for an experiment, and scientists look at your cells and they expose UV light to your cells, or they expose your cells to UV light, I should say. We call it XBA. Okay, UV light here. So these cells, your cells, will not demonstrate unscheduled DNA synthesis. And what unscheduled DNA synthesis is, it's a type of DNA synthesis that occurs as a result of nucleotide excision repair. So the UV light induces damage in the DNA, and nucleotide excision repair repairs that DNA and synthesizes DNA while it's repairing it. And unscheduled mean it's a type of DNA repair or synthesis that happens not during, it's not associated with the replication cycle. So it's happening at an unscheduled time. But so this XPA cell here is not is not synthesizing DNA because it's defective in nucleotide excision repair. So, however, let's go back to this case right here. So when we put XP1 with XP6 here, well, I can step back for a second. XP1 by itself, an XP1 cell by itself, if you expose it to UV light, you will not detect UDS. Same thing for XP6. So you expose that to UV light, you will not detect UDS. So no UDS. And how come? Okay, because we said XP6 is mutated in gene B, XP1 is mutated in gene A. Both of these are required for nucleotide excision repair, which manifests itself as unscheduled DNA synthesis. So neither of those have NER, so none of them can do UDS. A lot of acronyms here. So, okay, but when we fuse them and we expose the heterokaryon to UV light, well, UDS works now. Why? Because these nuclei, they complement one another. So XP1, if you remember, is mutated in gene A, but it has normal B. XP6 is mutated in gene B, but it has a normal A. So when they complement each other like this, I mean, this is called genetic complementation. So the normal phenotype is restored, and we call that complementation. So restoration of normal phenotype equals complementation. Okay, now I think we're ready to look at the question. Okay, let me make some room. Let me make some room down here. Okay, so here are all the different cell lines that scientists made heterokaryons of, and when they expose it to these cell lines, these heterokaryons, to UV light, they see some patterns here. So not all of them, not all of the heterokaryons performed UDS after exposure to UV light. So we can take a look at just right here. So if we take XP1, a cell line, and we fuse it with XP2 here, we can see, okay, a heterokaryon composed of XP1 and XP2 does not perform unscheduled DNA synthesis. Between XP1 and XP3 does not. How about between XP1 and XP4? Yes, it does. Okay, great. Okay, XP1 and XP5, yes. Okay, let's go down. Okay, so XP1 and XP6, yes. XP1 and XP7, yes. Okay, so 2 of the heterokaryons did not perform unscheduled DNA synthesis, and 4 of them did. So what does that mean? Okay, so it probably means, say 99% of the time here, is that this nucleus from the XP1 patient and the one from the XP2 patient in the heterokaryon are probably mutated in the same genes, right? So these are two different humans with xeroderma pigmentosum. You don't know why they have this genetic disorder. Any number of mutations could lead to this disorder. They could be missing gene A, they could be missing gene B, and by missing I mean just mutated homozygous recessive mutants or genotypes for these genes. We don't know, but we can perform this process and determine if they are mutated in the same gene or not. And it probably is that, you know, both of these individuals are lacking functional copies of A. So as a result, when you expose the heterokaryon to UV light, well, there's no functional A protein anywhere in the heterokaryon, so no NER and no UDS. Okay. However, if we form a different heterokaryon, let's get rid of XP2 here. Let's take XP4, the first one that worked. XP4, okay, well, UDS works here, so XP4 must have functional A's. And let's say XP4 is missing, let's say, C. Okay, he has 2, or she has 2 mutant C alleles for a third gene required for nucleotide excision repair. And XP1 most likely has 2 functional copies for this third gene. So 2 wild-type C alleles. So XP1 is making plenty of the C protein that is being shared by the whole heterokaryotic cell, and XP4 is making plenty of the A protein. Okay, so what does this mean? It means XP1 and XP4 complement each other with respect to UDS, unscheduled DNA synthesis. XP1 and XP2 do not complement each other. And we've shown, or we've discussed, the most likely reason when two genes do not complement each other in an experiment like this is probably because, they are the same gene. The mutants are in the same gene. And XP1 and XP4 complement each other because they are mutated in different genes. And as a result, when you mix them together, XP4 has functional copies of the gene XP1 lacks, and XP1 has functional copies of the gene that XP4 lacks. Okay, so we can look at, let me see here. So we have one more key thing to talk about here. So by doing an analysis like this, we can come up with a certain number of complementation groups. Complementation groups. And complementation groups usually represent numbers of genes involved in a process. So if there are 7 genes involved in a process, you should be able to, if you do a very thorough mutation analysis and complementation analysis, you should be able to find 7 different complementation groups. This is just a small analysis in this problem. There are more than the number of complementation groups involved in nucleotide excision repair and formation of or xeroderma pigmentosum disorder. But we have a certain number of complementation groups that we can see in this data over here. So to be in the same complementation group, you cannot complement one another. So what does that mean? Okay, so group 1, take all of the cell lines that did not complement each other in the assay. So XP1 and XP2 did not complement one another. XP2 and XP3 also, and XP1 and XP3 did not complement one another. So XP1, XP2, and XP3, any combination of heterokaryon between these did not complement one another. So these are in complementation group 1. Now to be in the same group, that means you do not complement each other with respect to the phenotype in question. So none of these complement one another with respect to unscheduled DNA synthesis. So let's take another group here. Let's go with group 2. Let's look at 5, 6, and 7. So 5, 6, and 7 do not complement. So if we make a heterokaryon between XP5 and XP6, they do not complement unscheduled DNA synthesis. XP5 and XP7 also do not complement. So we know XP5, XP6, and XP7 are all in group 2. And if we look at all the other combinations, say XP5 with XP1, yes, that complements. XP6 with XP2, yes. XP7 with XP3. You can see every other combination with XP5, XP6, and XP7 does complement. So only XP5, XP6, and XP7, different combinations between those, they never complement each other. So these are all in the same complementation group. So if you're in different complementation groups, if the genes are in different complementation groups, when you combine those, they will perform unscheduled DNA synthesis. For example, let's take a look at XP1 and XP5. So XP1 here with XP5, yes, okay, it does perform unscheduled DNA synthesis, as we would expect, because they're in different complementation groups. How about XP3 with XP6? So XP3 with XP6. Okay, perfect. Okay. Now we actually have and we can check every order and every combination, and we would find that every combination of mixing group 1 with group 2 complements unscheduled DNA synthesis, or the lack of unscheduled DNA synthesis. It restores it in the heterokaryon. We have one more group, and it's a lonely group, and it's just XP4. So take a look at XP4. So XP4, when you combine it with everything, XP1, we combine it with XP1, yes, complements XP1, complements XP2. When I say complements XP2, I mean it complements unscheduled DNA synthesis. When combined into a heterokaryon with XP2. XP3 complemented. Okay, now we have XP4 with XP5 complemented. XP4 with XP6 is complemented. XP4 with XP7 is complemented. So XP4 complements everything except for itself right here. So if you were to take two XP4 cells and just fuse them together, you would not see unscheduled DNA synthesis, and that makes sense. So XP4 is in a group by itself here. So we have three different complementation groups, and the most likely explanation for the existence of three complementation groups in this data is that group 1 represents a gene that's required for unscheduled DNA synthesis, and we're going to call this gene A, and all of these individuals right here are carrying recessive alleles for gene A. They're mutant, they don't work. Group 2 is another gene, I think we called it gene C. All of these individuals carry mutant versions of gene C, and so that's why they have, their cells can't perform unscheduled DNA synthesis, and it's why they have xeroderma pigmentosum. And group 3 is a different gene, we can call this one gene B. And the XP4 individuals are mutated in a third gene, carrying two mutant alleles for the protein required for nucleotide excision repair and unscheduled DNA synthesis. So we discovered at least 3 genes through this process, and I think scientists have discovered like 7 using the similar technique. So 7 or more, I don't know the exact number, but many different genes required for xeroderma or for nucleotide excision repair. And so if you do this type of analysis, you can find many different complementation groups, and without even knowing the nature of the mutations or the exact genes that are mutated, you can determine approximately how many genes are involved in nucleotide excision repair. Okay, that was okay. Again, if you're a little confused, take a look at lecture slides 16. There are 4 or 5 slides towards the end of the lecture slides that will walk you through this problem in a written form.